Hello and welcome to Toddcast number 49. This ended up being the last of our free radical Toddcast. And the reality, this one I hope is the most interesting of all because it applies to some of the things we see in biology. But when we talk about free radicals in organic chemistry, we talk about a highly reactive species, an unpaired electron that wants to combine with anything and everything. And in our world that surrounds us, we have mostly nitrogen gas, triple bond between two nitrogens, lone pair on each nitrogen, nice and stable. But we also have O2. About 22% or so of our atmosphere is made up of O2. And O2 is not actually a double bond between O's like they sometimes show you in GChem. It's actually a biradical. Now, this is based on molecular orbital theory, and so you can argue whether that's valid or invalid, but the point is, it explains the reactivity. Radicals are highly reactive. O2 is highly reactive. It can literally burn anything, including what should be a highly stable alkane. Alkanes really don't do much of anything. We can brominate them and chlorinate them, as we learn, but even then, they don't really do that much. And so reaction with O2, that's about it. They don't do much else. So they only do radical reactivity in the with O2 because O2 is a radical. Okay, so in our body, because we breathe in O2, we want it to break down the right molecules in our body. We want it to break down fuel molecules so we can get the energy out of them. So the whole burning, the whole oxidation process, we want it to be in our favor, generate energy, break down fuel molecules, kick out CO2 and water, everything's fine. The problem with this is O2, being so reactive, can start reacting with everything in our body. So we are built to survive. The free radicals that form, we have certain scavenger molecules naturally occurring in our body that pick up free radicals. And it ends up, the SH, the thiol functional group, is actually really good at carrying out hydrogen extraction, meaning it can put an H on that free radical and have a stable S free radical that can then combine with another S, form a cross-link, and then that can be disposed of, or it can be reduced back down to being thiols again. So an SH, because of its ability to form an SS bond, disulfide linkage, can act in a way to donate H's through a free radical process and pick things up. So this molecule, things being free radicals that end up in our body. So this molecule, it's important to have at least a basic idea of amino acid chemistry. And amino acids make up, there's subunits of our proteins. So if you look for these linkages right here, that's a peptide linkage, also known in chemistry as an amide bond, but a peptide linkage. And I look here at this peptide linkage. That means I have three amino acids. I have this first one here, I have this second one, and this third one. The interesting thing about this first amino acid is that it's actually forming a cross-link, or I should say the next bond over, not using its normal C-terminal, but instead using its side chain. This hopefully will make sense in a second, but if we consider what these used to be, there used to be an OH here and an H here, used to be an OH here and an H here. So if we spread these out, it will turn into this. So let me get rid of my links. And notice that OH here and this H here, that used to be a bond N. OH here and this H that went, and that used to be a bond to N. So I have three amino acids. You don't have to memorize for this class, but knowing them certainly doesn't hurt. So this first amino acid is glutamic acid. Here's my carboxyl terminal, my N terminal, and the side chain is CH2, CH2, and an acid. Its symbol is GLU, or one letter code, or shorthand is E. Cysteine is going to be very, very common when you take bio, amino terminal, carboxyl terminal, and the side chain CH2SH. Ends up this one is very useful in terms of cross-linking, like we just mentioned, forms disulfide linkages, which affects protein structure. And then the last one is glycine, the simplest of all. Side chain is an H. So there's an H like there normally is. There's an unwritten H here, unwritten H here. There's an H here, which is both a side chain and the unwritten one. Okay, but these three amino acids combine to form this antioxidant. Very common, it's one we generate, and again, it can deliver an H for hydrogen extraction, make an S free radical which binds with another one, then you reduce it back down, and another reaction, and starts all over. Okay, so it's a thiol group that is so useful for hydrogen abstraction. Okay, useful reactions biologically. One of the cooler things is how our body responds to instantaneous impact, pain, cuts. We have our immune system, and one of our immune responses is instantaneous at the cellular level. So we have hormones that are released from glands, they travel through the body and send messages. But if you place your thumb onto a sharp object and let's say cut your thumb open, you probably want to not keep your thumb there. The whole idea of continually putting your thumb on a sharp object would be bad for our longevity. So upon contact, among puncturing, a handful of things happen. A pain response, a repair response, thrombin, and then a I'm not sure, just overall like cell-to-cell communication of get rid of the dead cells. So I guess that is kind of repair as well. But here's how it happens. In our cell, in our individual cells, in the cell membrane, we have arachidonate. And when triggered, when exposed to O2, enzyme will kick in and cause this to turn into a series of prostaglandins, what's known as the cyclooxygenase pathway. There's actually two, COX-1 and COX-2 enzymes, where they will convert this ring system into this more complex prostaglandin G2. The 2 here stands for 2 double bonds. G just happens to be this particular structure, and it's the first of 2 intermediates. So arachidonate will go to prostaglandin G2, which eventually will go to prostaglandin H2. So here's the arachidonate, turns into prostaglandin G2, and then follows the following mechanism. So again, as mentioned, oxygen is a biradical. So oxygen is going to react here apparently, and here apparently, at carbons 9 and 11, and carbon 15 down here. So double bonds can move around, but I want to look at this one first. This radical can go here, this radical can go there, this radical goes there, that one go there, which means you have a radical that can go from here and here, resulting in a ring that's 1, 2, 3, 4, 5, like it is here, 1, 2, 3, 4, and 5, with an O coming from what is carbon 9 and carbon 11 in the overall 20 carbon species, so 9 to 11. And then this can now be functionalized in so many different ways to turn into different things that can send different messages. But this overall 20 carbon fatty acid can be used as a precursor to generate different messenger molecules. It's also going to react at the allylic position. So notice this 1, 2, 3, 4, 5, 6, 4, 5, 6. So right here, we're going to have that same sort of issue where this free radical is going to go on, get an oxygen. And my double bond ends up moving over. I do hydrogen extraction somewhere else, but we'll see that as we go. So there's the blue arrows. Let me get rid of what I've drawn. So it shows this ring formation here. Notice it is a syn addition at first. The 2 O's are going at the same time, so they have to go to the same side. So this goes on here, this goes on here. We have these 2 processes, and it continues on to PgH2. So the second one after the 2 O2s, one adds to this system, one adds to here. This O2 basically inserts into the CH bond and the double bond moves over, so it's a free radical insertion. You end up with this. We now reduce this down, and this functional group is free to go on to form a bunch of different other things. Not going to get into the mechanism of that, but through a series of oxidation reactions, you can turn this carbonyl into an OH. Fairly simple. So this one is for pain. So this forms very, very quickly. There's also one for clotting. So this does a little bit different thing. So you notice this O actually inserts into the ring while this one stays bound to it. So you still have carbon number 9 and number 11, but this O inserts into the bond between 11 and 12. Notice the rest of the prostaglandin is very similar, but this portion being different than this sends a different message, binds receptors differently, and this one calls for clotting. Prostaglandin chemistry is massive. There is no rhyme or reason why we call this A, E, G, or H chemically. It's just historically how they were discovered. So when they started discovering there was more than one type of prostaglandin, they said, okay, let's call the first one A, second one B, third one C, which means historically we discovered the prostaglandins that act before discovering the pathway that made them. And so somewhere along the line, they discovered this prostaglandin G2, which was a precursor. So we knew what prostaglandins were, discovered what it was, and then later discovered, oh, hey, there's a second step. And then these go on. So it's not a bioclass, so I don't want to go into too much detail on these. But simultaneously, it's incredibly interesting how these reactions can happen so fast and with enzymes that make them so specific that you can make a molecule that can send almost instantly the message for pain, for clotting, for swelling and repair, for whatever we need to do. And so I guess what I'm starting out with, clotting just in forming the coagulant to stop the bleeding, and the repair is actually generating new cells and knowing how to get rid of the dead cells. So again, just this concept of a biradical reacting with one of our molecules quickly and ending up with this prostaglandin chemistry. So hopefully this is interesting and brings somewhat interesting light to the concepts here. But useful radical reactions, last of the ones we want to bring up, antioxidants. We mentioned early on, I think probably first or second lecture, that phenols, or in this case, 2 OHs on this. So if they're 1 and 2, positions 1 and 2 are known as ortho, they're known as catechols. 1, 4, I'm just going to say 2 hydroxys on a benzene ring. This compound, a bunch of different ways it can react, but one of them it can do is give away an H free radical as necessary. So it too is a free radical scavenger. This functional group you'll see in a lot of naturally occurring foods we bring in to help act as antioxidants. So if you extract the H off, so something comes in. I'm just going to say some free radical comes in, extracts the H, and with this free radical here, it can resonate over. So this one can go here, this one go here, this one, and end up with a free radical down in this position right here, which means this is once again free to react with an H. And you end up with this compound, meaning it can take care of 2 free radical hydrogen extractions in going from what's known as hydroxyquinone or hydroquinone to quinone. This is a biochemistry reaction that happens to be rooted in organic chemistry knowledge. So I expect you to have a general idea of what functional groups the SH and the OH on a benzene ring. So the SH by itself, the OH on a benzene ring, are useful as free radical scavenger reactions. So knowing what aspects of OChem can explain biochemistry, that's the most useful thing you'll get out of this class from the majority. If you're moving on as a chem major, the polymerization is perhaps more interesting, molecular design, the synthesis. If you're moving into some bio-related field, the prostaglandins, antioxidants, and then particularly the quinone and hydroquinone. Hopefully this sums up free radicals. They're somewhat interesting. For this chapter, make sure you know how to identify selectivity rules, how to use the math. That's going to be key. You know, a few reactions here and there, and just have fun with it. So thank you once again for joining in, and I will stop the share and see you soon. Bye.